Hello and good morning. I'm happy to have the opportunity to present our work on behalf of my colleagues here today. And I also would like to thank the organizers for asking for an oral presentation of our abstract on how tamoxifen instigates uterine cancer development. Tamoxifen was first discovered in 1967 and is the first hormonal drug to be approved for the treatment of ER-positive breast cancer. Today, tamoxifen is used worldwide for the treatment of ER-positive early-stage disease and significantly improves overall survival. However, one serious drawback of tamoxifen treatment is that tamoxifen increases the risk of uterine cancer development. Previous studies have reported a 2 to 7-fold increased risk after 2 to 5 years of tamoxifen treatment. More recent results from the ATLAS study showed that the risk increases with a longer duration of tamoxifen use and that there is a 2-fold further increased risk at 10 years compared to 5 years of treatment. So in this study, we aimed to understand the mechanisms by which tamoxifen increases the risk of uterine cancer and to identify potential therapeutic approaches to prevent tamoxifen-associated uterine cancer in the future. In the typical model of therapy-related cancerogenesis, as shown here, cells are exposed to drugs, such as chemotherapy, that promote cancer development and progression. These cells may acquire driver mutations that are caused or selected by these drugs, which then can result in clonal expansion and ultimately cancer. We therefore asked whether this model also applies to tamoxifen-associated uterine cancers. To answer this question, we explored their genomic landscape. We used samples from the TAMARIS study, a Dutch cohort studies on uterine malignancies in women with breast cancer. Our discovery cohort consisted of 21 tumor normal pairs in which we performed whole exome sequencing. We then validated our results using an independent set of 40 tamoxifen-associated uterine cancers in which we performed ddPCR. These are the details of tamoxifen therapy in the whole exome sequencing cohort. The median duration of tamoxifen use was around 2 years, and the majority of patients was diagnosed within 1 year of last tamoxifen use. In the next part of my presentation, I will show you our results. Throughout the figures that I'm going to present, tamoxifen-associated uterine cancers are abbreviated as TAUCs and denoted in purple. We used a control cohort of more than 540 de novo uterine cancers from TCGA that are not associated with tamoxifen use, and they are denoted in blue. So we first looked at the surrogate of molecular subtypes in uterine cancer using the whole exome sequencing data and found all 4 known subtypes in the de novo uterine cancer cohort, that is POL-E, MSI, chromosomal instable and stable samples. We then compared them to the molecular subtypes in the tamoxifen-associated uterine cancer cohort and found all 4 molecular subtypes to be present in that cohort and with equal frequency, highlighting the quality of our data set. Next, we analyzed driver gene mutations. As you can see in this cohort plot, we identified 4 significant genes: PTEN, KRAS, TP53, and ARADA1. To increase the statistical power, we also restricted our analysis to 140 known uterine cancer genes and identified 3 additional driver genes, as you can see here. All of these driver genes were reported previously as drivers of de novo uterine cancer and are highly prevalent in that cancer type. We next wanted to see whether there is a difference in mutational frequency between the de novo uterine cancers, depicted on the left, and the tamoxifen-associated uterine cancers, shown on the right. We looked only at genes where we were powered to detect differences, as depicted here, and found 2 such genes that had a lower mutation frequency in the tamoxifen-associated uterine cancers. We saw only 3 PIK3CA mutations and no single PIK3R1 mutation. And interestingly, both of these genes are key components of the PI3 kinase pathway. So we then tested for significance and indeed found a significantly decreased number of mutations in both of these genes, in tamoxifen-associated cancer. Next, we asked ourselves, is it truly fewer genomic changes or only fewer mutational events? So we included copy number changes as shown on the right and still identified a difference. So we then took an independent validation set of 40 tamoxifen-associated uterine cancers and analyzed PIK3CA hotspot mutations using ddPCR. Again, the PIK3CA mutation frequency was significantly lower in the tamoxifen-associated uterine cancers than in the de novo cancers from GENIE, validating our findings from whole exome sequencing. So together, our genomic analysis revealed a decrease in the frequency of PIK3CA and PIK3R1 mutations in tamoxifen-associated uterine cancer. Based on this, we hypothesized that tamoxifen might substitute for the mutant-driven activation of the PI3 kinase pathway. This hypothesis is supported by previous studies that have shown a crosstalk between ER and the PI3 kinase pathway in breast and uterine tissue involving IGF-1. So now here we wanted to know whether tamoxifen can activate this ER-PI3 kinase crosstalk in normal uterine tissue, and whether it can be specifically abrogated by a PI3 kinase inhibitor. To answer this question, we turned to an in vivo mouse study in harvested uterine horns from mice that were randomized into 3 arms: control, tamoxifen treatment, and tamoxifen treatment plus the PI3 kinase inhibitor alpelisib. So we first looked at some of the key targets of the PI3 kinase signaling pathway, as shown in these EHC figures of uterine tissue here, and found indeed that tamoxifen can increase the activation of the PI3 kinase pathway that was further then abrogated and specifically abrogated by the PI3 kinase inhibitor alpelisib. So we then asked, how is IGF-1R activated? RNA sequencing of luminal cells showed a decrease in IGF-BP3, both with estrogen and tamoxifen, but no significant changes in IGF and IGF-1 receptors. We then looked at the stroma using RNAscope and saw a significant increase in IGF-1 with tamoxifen treatment that was reversed by alpelisib and is suggestive of a paracrine effect. So together, our data suggest that tamoxifen can indeed substitute for the mutant-driven activation of the PI3 kinase pathway. Since our preclinical results showed that the tamoxifen-induced activation of the PI3 kinase pathway is an early event, we went back to our genomic data to see whether the early genomic events in tamoxifen-associated uterine cancers are different from the ones in the de novo uterine cancers. And indeed, we found that tamoxifen-associated uterine cancer was missing 1 early clonal driver event compared to the de novo uterine cancers. To further investigate whether this missing early event is potentially a PIK3CA mutation, we turned to the 3 tamoxifen-associated cancers that did have a PIK3CA mutation and that are shown here. So we found that in all 3 samples with the PIK3CA mutations, this mutation was among the early clonal events. And this suggests that in these samples, the PIK3CA mutations may have occurred prior to tamoxifen treatment, and that PIK3CA mutations are typically relatively early events and maybe the one missing event in tamoxifen-associated uterine cancer. So with this, I slowly want to come to an end and summarize the key findings of our study. Our results highlight the mechanistic differences in the early events that lead to the development of tamoxifen-associated uterine cancer. Our findings also offer a new mechanism of therapy-related cancerogenesis in which a drug activates a signaling pathway that substitutes for the selection of a driver mutation. Our results are in line with clinical findings, implying that the increased risk of uterine cancer is confined to the duration of and a short period after tamoxifen treatment. Our results may also provide future therapeutic approaches to prevent tamoxifen-associated uterine cancer. One limitation of our study, however, was that the number of samples is relatively small, since tamoxifen-associated uterine cancer is not a common disease. Before I stop, I want to thank the many people who contributed to the study. Especially, I want to thank Renate and Geddy for the immense support and help, Agustina, who did all the in vivo experiments, Wilbert for making available the study cohort, and Yossi for his invaluable insights. I also want to thank the patients and the funding source, and thank you for your attention, and I'm happy to take questions.